Hi, my name is Ramesh, and I will be presenting on the impact of a mutation in fatty acid uptake within our cells. An imbalance of fatty acids can cause obesity, diabetes, cancer, and cardiovascular, neurodegenerative, autoimmune, and liver diseases. In this study, a single point mutation was created in a protein called cluster of differentiation 36, or CD36. Lysine, which is a positively charged amino acid, was replaced by glutamine, a polar non-charged amino acid. So this mutation would result in a significant decrease in fatty acid uptake within our cells. It also causes a change in the amino acid charge, altering and maybe even inhibiting the protein's function. In this case, pharmaceuticals can then be created in order to decrease fatty acid uptake by targeting this particular residue. A long-chain fatty acid is composed of a carboxylic acid that contains a long aliphatic hydrocarbon chain. Long-chain fatty acids can be transported into the cell through a passive diffusion flip-flop mechanism. However, over recent years, it's been discovered that they can also enter the cell through a protein-mediated mechanism in which they require the help of a specific protein. This is the CD36 protein we were talking about earlier. Here is a simplified structure of CD36 showing its two transmembrane domains. CD36 has a transmembrane channel that contains two extracellular entrances where the fatty acids can enter the cell from. Our hypothesis is that fatty acids move through the membrane by cross-linking to the lysine residue at position 164, close to the phenylalanine residue that's circled here in red, entering the central cavity and then making their way into the cell membrane surface. So, where is CD36 found in our body? CD36 is found in our blood cells, immune cells, fat tissues, kidney cells, bone cells, and even endothelial cells. The research aims of the project are as follows: to determine if the mutation only affects fatty acid uptake and does not disrupt the CD36 signaling, and to identify whether the mutation causes a decrease in the fatty acid uptake of our cells. We studied the research aims by three means: DNA isolation, a fatty acid uptake assay, and a seep assay. Mouse DNA samples were purchased and their identification was confirmed by using the DNA Sanger sequencing method. The CD36 protein's concentration, purity, and the site of mutation was also confirmed. Cells were purchased and grown in tissue culture plates with highly concentrated glucose. The mice DNA samples that we just talked about were then transfected into the cells, and after the cells were 80% grown, they were transferred to 12-well plates, as shown in the slide, and the GFP expression was assessed. GFP expression measures the expression of CD36 within the cell. Once cells were 80% grown, they had primary antibodies, which is shown in red, and secondary antibodies, which is shown in blue, added to them to assess immunofluorescence. Immunofluorescence allowed us to measure the CD36 expression within the cells. Cells were once again purchased and grown on tissue culture plates. Once transferred to 12-well plates, a bacterium was added to the samples. A seep assay was then carried out in a 96-well plate. Seep is a reporter gene that measures the level of cell signaling, and seep expression helped us determine whether this mutation that we had would result in a dysfunction of the CD36 signaling because of the bacterium. So, if cell signaling was disrupted, lower seep expressions would be observed. DNA identification, purity, and absorption were all successful. The wild type and the KQ mutant plasmid sequences were also confirmed. The cells made connections with one another and started growing with no sign of contamination. Cells were then looked at under the microscope to detect GFP expression, which, as I said before, it detects the amount of CD36 protein. Both wild type and the KQ mutant fluoresced green, while, as expected, non-transfected cells did not. Additionally, individual cells showed bodies and glowed green around a blob in the cell rather than showing it on the cell membrane. Both wild type and the KQ mutant showed the presence of CD36 receptors. They fluoresced green and red. Non-transfected cells had not been transfected with CD36, so they didn't fluoresce under GFP. They didn't show any signs of green or under immunofluorescence. Wild type also fluoresced at higher brightness than the KQ mutant did. If signaling was disrupted, cells would express a very low CEAP. This would also depend on the number of cells alive or the number of cells showing GFP. Because the KQLs had a lot less cells present, that's shown in lighter blue, the amount of GFP fluorescence was lower in comparison to wild type. Non-transfected cells were also used as controls and expressed no GFP and no CEAP. CEAP expression was the greatest in wild type, but this was only because wild type had more cells. So we had to make ratios. So if you can see, there were 6.8 times more cells in wild type compared to KQ, while the CEAP expression was only 1.5 times greater. Since CEAP measures the signaling of CD36 and its levels were much lower, it suggests that CD36 signaling was not disrupted. CD36 is a significant signaling molecule, as I've mentioned before. The mutation we created replaces a positively charged lysine residue with a neutral amino acid, glutamine. When fatty acids from our diet are transported into the cell, they bind to the lysine residue on the CD36 protein, which is shown as this blue-pink protein on the cell membrane. They then pass the cell membrane through one of CD36's channel. If the lysine residue is changed to include glutamine, the fatty acid would be unable to bind and translocate into the cell. Replacing lysine with glutamine can also change other functions of CD36's protein. So if we want to conclude that it is this part of the protein that is responsible for fatty acid uptake, the rest of the protein's functions, such as its signaling, must be kept intact. CEAP expressions were low, meaning that our mutation did not disrupt signaling and CD36 was in fact functional. The mutation K164Q was expected to cause a loss of fatty acid uptake, while at the same time preserving other CD36 functions. But because we didn't have much time, the only conclusion we can make is that the KQ mutation does not disrupt CD36 cell signaling. So we don't know whether fatty acid uptake was increased or decreased because of this mutation. Immunofluorescence was brighter in wild type compared to the K164Q mutation, suggesting that the mutation may have caused changes in the CD36 receptor, but this would need to be observed further with a fatty acid uptake Bo-Dippy experiment. CD36's function in fatty acid uptake plays a critical role in obesity, diabetes, heart diseases, stroke, cancer, and many brain and autoimmune diseases. In this study, the KQ mutation in CD36 was predicted to decrease fatty acid uptake, but also at the same time retain other important CD36 functions. But because we didn't have much time, we could only complete the CEAP assay and conclude that the mutation does not cause a disruption in CD36's normal signaling. Thank you so much for listening to my presentation.